I welcome you to this biology paper 2 2022 GCE representation. So if you are new to this channel, kindly subscribe for more videos and share the video if you find it useful to you, to your friends. Okay, so I'm going to answer section A, all the questions from section A. Then as for section A, it carries 44 marks, so which means you need to get all these 44 marks. Okay, so the question is saying, which is question 1. Figure 1 shows cell division in animal cells. Figure 1 shows cell division in animal cells. So now we have the zygote. We have the zygote. So the zygote divides by forming cell A and cell B, which are identical. As you can see, they are identical. Even the shapes they look similar. Now as for cell A undergoes some specific changes to form a muscle cell. Now this process of cell A changing into a muscle cell, we call this one as cell differentiation. We call this one as the cell differentiation or cell specialization. So now during cell division, cells are produced in numbers and they look similar. Like as you can see, cell A and cell B, they look similar, but during cell specialization or cell differentiation, each cell is going to have its shape in order to carry the specific function. So as for cell A has changed to a muscle cell, so we can call this process as the cell differentiation or cell specialization. Okay, then as for cell B divides to form two daughter cells which are diploid. So when you look at cell B looks similar to the two cells that cell B has produced, the new cells. Then as for these new cells, they are also identical in terms of the shape and the way they are looking. Okay, so we can say the process of cell B to change to form new cells, this one is called mitosis, because during mitosis, two daughter cells which are diploid and they are genetically identical, they are produced. Okay, so I'm saying cell B has changed to form two cells which are identical and they are diploid in number. Okay, so now the first question is saying, which is question A, Roman numeral number 1 is saying, state the characteristic of living organisms being demonstrated in figure 1. Of course, this one here, you can think of reproduction. Now, this one here, it doesn't involve the fusion and the fusion of the male gamete and the female gamete. So this one here is not going to be the production, but it is going to be the growth. Okay, so as for the growth, it is the permanent increase in the size and the mass of an object. So here I'm saying this one here, the answer of question A, Roman numeral number 1 is growth. This is because as for growth is the permanent increase in size and the mass of an organism or the body of an organism. So now as you can see, the zygote divide to form two cells which is A and B. They are dividing and they are growing, which means this one here is one of the characteristics of a living organism, which is the growth. So the answer for question A, Roman numeral number 1 is the growth. Then as for Roman numeral number 2 is saying, explain what has happened to cell M. Explain what has happened to cell M. So now as you can see on this diagram here, cell A undergoes cell specialization to form muscle cell. So I'm going to say cell A undergoes a cell specialization to form a muscle cell or to form the muscle cell. So cell A undergoes cell specialization, or you may say cell differentiation to form the muscle cell. Then apart from that one, we also have question B, which is saying, explain how the zygote in figure 1 can result into the formation of an organism through cell differentiation. Okay, so now here I'm going to say the first thing you're going to find that the male gamete, which is the sperm, and the female gamete, which is the ovum, they are going to join, or they are going to fuse. Let me say to fuse. So the male gamete and the female gamete, they fuse. Okay, after fusing, we are going to find that a zygote is going to be formed, which is this one in the diagram. Okay, then from there now, a zygote is going to develop into an embryo. A zygote is going to be developed into an embryo. A zygote is going to be developed into an embryo. Then an embryo is going to be developed into a fetus, or we can say fetus. Some they say fetus. Okay, so the zygote is going to be developed into an embryo. Then an embryo is going to be developed into the fetus. Then the last one, an organism is going to be produced, which is a baby. So here I was saying the male gamete and the female gamete, they fuse to form a zygote. Then the zygote develops to form an embryo. Then an embryo develops to form a fetus. Then the last one, a fetus now develops into a baby, which is an organism. Okay, so that one is 3 marks. Then as for question C is saying, describe two life processes of living organisms and state the law of enzymes in the two processes. On this question here, they are not asking you to name the enzymes, but they are asking you to give them the life processes and also the law of enzymes, or the role that enzymes play in those life processes. So the first one, I'm going to say nutrition. Now, the nutrition, I'm going to give an example of photosynthesis. I'm going to give an example of photosynthesis. Now, during photosynthesis, we are going to find that small molecules are going to be synthesized. Okay, so small molecules are going to be synthesized. So an example that I can give is the synthesis of proteins, the synthesis of proteins or amino acids. So here I'm saying an example is photosynthesis under nutrition. So a life process here, it is nutrition. Then an example, it is the photosynthesis. Now, under photosynthesis, we are going to find that these small molecules are being built up to form a large molecule. Okay, so I'm going to say the law of an enzyme, or the law of enzymes here, is to build up small molecules into bigger molecules. An example, synthesis of proteins or amino acids. Then the next one, I'm going to say respiration. Respiration. Now, during respiration, we are going to find that the law of enzymes here is to break down now large molecules into smaller molecules. So I'm going to say respiration. Then the law of enzymes is to break down large molecules to form smaller molecules, to break down large molecules to form smaller molecules. So that is the last one for question 1. Let's move on to the next one, which is question 2. Okay, so question 2 is, this one is an internal structure of a leaf. So the question is saying figure 2 shows the transverse section of a leaf. Figure 2 shows the transverse section of a leaf. So we have X, we have structure X, structure Y, structure Q, and structure R. So now, before I answer the questions, let me try to label the structures. So as for structure X is called the upper epidermis. Structure X is called the upper epidermis. Then as for Y is called the palisade cell or palisade tissue. Y is called the palisade cell or palisade tissue. Then as for Q, this Q here, we are going to consider it to be a vesicular bundle. Q is going to be the vesicular bundle. Q is going to be the vesicular bundle. Open bracket, we can say xylem, phloem. Open bracket, we can say xylem, phloem. Then as for R, this R here, it is called the Agadi cell. Cell R is called the Agadi cell. Okay, so we have the carbon dioxide coming out. Let me say we have carbon dioxide diffusing through the stomata or the guard cells. Then we also have the water vapor coming out from the leaf through the stomata pore. Okay, so now the first question, which is question A, Roman numeral number 1, is saying identify tissues X, Y, and D, X, Y, and D, Q. So now as for X, I was saying X is called D, the upper epidermis. X is called D, the upper epidermis. Then as for Y, Y is called D, the upper, let me say, Y is called D, the palisade D tissue. So in this case here, since they are saying identify tissues, so I'm going to say X is called the upper epidermis tissue. The upper epidermis tissue. Then as for Y, I'm going to say palisade D tissue. Then as for Y, I'm going to say palisade D tissue. Okay. Then as for Q, I'm going to say vesicular bundles. Vesicular bundles, open bracket, X, let me say xylem, phloem, let me say xylem and phloem tissues. As for Q, I'm saying vesicular bundles, open bracket, xylem and phloem tissues. Okay, the next one, which is Roman numeral number 2, is saying tissues X, which is the upper epidermis tissue, and Y, which is the palisade D cell or palisade tissue. Now, as you can see on the palisade D tissue here, we have the abundance of chloroplast, or I may say chlorophyll. If you don't want to use the term of chlorophyll, you can use the chloroplast. So as for Y, contains the abundance of chlorophyll or chloroplast. So tissues X and Y were tested for starch. So now when you're testing for starch, or when you're testing a leaf for starch, when starch is present, the color turns blue, black. Okay, so when it is absent, the color turns brown or yellow. So now in this case here, they are saying tissues X and Y were tested for starch. Which of the two tissues would show the presence of a high concentration of starch? Now, as you can see here, tissue Y is having a lot of chloroplast, or I may say chlorophyll. Now as for tissue X is not having chloroplast, or it is having little chloroplast or chlorophyll. So which means when we are testing, when we take tissue X and tissue Y for testing, when testing for starch, we are going to find that tissue Y is going to show positive result, but tissue X is going to show a negative result. So here I'm going to say Y. Here I'm going to say tissue Y, because tissue Y contains the maximum chloroplast or maximum chlorophyll. Okay, so the next one, which is Roman numeral number 3, is saying give an explanation for the answer given in A, Roman numeral number 2, okay, which is 3 marks. So here I was saying for Roman numeral number 2, I was saying it is tissue Y, because this one, it is a palisade cell and it contains maximum chloroplast or abundance chlorophyll or chloroplast. So now we need to give a reason why we are saying this one is going to show concentration of starch, which is the structure Y. So now the first one, I'm going to say because of maximum chlorophyll or chloroplast. Because of maximum chlorophyll or chloroplast. Then the next one, or let me repeat again. Here I'm saying the presence of maximum chlorophyll or chloroplast. Then the next one, I'm going to say the site for photosynthesis. The site for photosynthesis. The site for photosynthesis. Apart from that one, I'm going to say the site for respiration. The site for respiration. Okay, so those are the answers for question A, Roman numeral number 3. So let's move on to the next one, which is B. Okay, which is B. So B is saying, which is Roman numeral number 1, it is saying name the cell labeled R in figure 2. So like the way I was saying, R is called the guard cell. R is called the guard cell. So my answer here is going to be guard cell. Then as for Roman numeral number 2 is saying, which feature does cell R have in common with tissue X and Y. That enables it to perform its function. Now, as you can see here, tissue Y is having chloroplast or chlorophyll. Even tissue R is having chloroplast or chlorophyll. So let me point at the chlorophyll. The chlorophyll are these, or chloroplasts are these. Okay. So now here I'm going to say the presence of chlorophyll in both tissue Z, X and Y. The presence of chloroplast or chlorophyll in both tissue Z, X and Y. So that was the last one for question 2. Let's move on to the next one, which is question 3. So question 3 reads, Figure 3 represents bones of a forelimb of a mammal. Figure 3 represents bones of a forelimb of a mammal. Figure 3 represents bones of a forelimb of a mammal. So we have structure labeled I, J, K and H. Now before I answer on this part here, let me try to label the parts, the structures or the parts. So as for structure I, this structure I, it is called the humerus. Structure I, it is called the humerus. Structure I is called the humerus. Then as for structure J, this structure J, it is called the synovial joint. Now specifically, it is called the hinge joint. I'm saying structure J, it is a synovial joint. Now specifically, it is the hinge joint. Okay. Then we have structure H and K. Now on this part here, I'm going to help you, or now you are going to differentiate between ulna and radius. So I'm going to say ulna is longer than the radius. Ulna is longer than the radius, which means the longest bone here between K and H, it is K. So I'm going to say K is going to be my ulna. Then as for H is going to be my radius. I'm saying K, which is ulna. Ulna is longer than the radius, or we can say radius is shorter than ulna, which means ulna is longer than the radius. Okay. Apart from that one, you need also to look at, you need to know the bone disorder or disorder of the joint at J. So the disorders at J, the disorders of the joint at J, we can talk of bone dislocation, or you can say gout. Bone dislocation and gout. Okay, so let's answer the questions. So question A is saying, which letters correspond to the following structures? Which letters correspond to the following structures? So we have Roman numeral number 1, which is the radius. So I was saying radius is shorter than the ulna. So radius is going to be H. Then Roman numeral number 2 is saying ulna. Here I was saying ulna is longer than the radius, which means K is going to be our ulna. Then as for Roman numeral number 3, it is saying humerus. Humerus is represented by capital letter I. Okay, so I is our humerus. Then as for B, Roman numeral number 1 is saying identify the type of joint found at J. Identify the type of joint found at J. So I'm going to say the synovial joint, open bracket, hinge joint. So here I'll say specifically this one here, it is the hinge joint, but it is a synovial joint. Okay, so on the synovial joint we have two types of synovial joint. We have the ball and socket joint and also the hinge joint, but the one which is shown on the diagram here, which is J, it is called the hinge joint. So I'm going to say synovial joint, open bracket, hinge joint. Then Roman numeral number 2 is saying give a reason for your answer in B, Roman numeral number 1. So I'm going to say, as you can see, this one here, it is the forelimb, it is the arm. Okay, so now as you can see, when you straighten the arm, you're going to find that the arm is going to move only on one plane. Okay, so when you straighten the arm, you're going to find that the arm is going to move only on one plane, which is the 180 degrees. Okay, so I'm going to say this is because it moves only on one plane. This is because it moves only on one plane. This is because it moves only on one plane. Okay, then as for question C is saying name two disorders of the joint at J. So name 2 disorders of the joint at J. So J is the in joint. So the disorders that J is likely to be affected to is the first one I'm going to say bone dislocation. I'm going to say bone dislocation, followed by the gout. Okay, followed by the gout. So that one is the last one. Okay, so let's move on to the next one, which is question D. Question D is saying, explain why a bone is considered to be a living tissue. 2 marks. Explain why a bone is considered to be a living tissue. 2 marks. Okay, I'm going to say a bone is considered to be a living tissue because it contains living cells. Because it contains living cells. For example, red blood cells, white blood cells, platelets, and so on. Okay, so here I'm saying a bone is considered to be a living tissue because it contains living cells. Okay, it contains living cells. The first one is the red blood cell, the white blood cells, and the platelets. Apart from that one, the bone is considered to be a living tissue because it contains blood vessels. It contains blood vessels. So in the bones, you are going to find that a bone contains blood vessels which supply blood. Okay. Apart from that one, a bone is considered to be a living tissue because it can be affected by bone marrow cancer. So this bone marrow cancer affects the what? The bones. Okay. So the next one is question 4 is saying, but before I move on to question 4, let me repeat. A bone is considered to be a living tissue because it contains living cells. Now, those living cells, we have the red blood cells, white blood cells, platelets, and so on. Apart from that one, blood is considered to be a living tissue because it contains the blood vessels. It contains the arteries, those things, yeah, like that. Apart from that one, this a bone is considered to be a living tissue because it can be affected by bone marrow cancer. Okay, so this bone marrow cancer affects the bone marrow. Yeah. Then as for question 4, question 4 is saying figure 4 shows let me say schematic diagram indicating the movement of water in a plant from the roots to the leaves. Okay, so figure 4 shows a schematic diagram indicating the movement of water in a plant from the roots to the leaves. Okay, so on this part here we have the substance which is coming from the leaves, which is O, and also we have structure M, which is showing the movement of water from the roots to the leaves. Okay, then we have also structure P, and also the movement of water in the root air cell, which is denoted by capital letter L. So now, before I answer the questions, let me try to explain a little bit on this part here. Sometimes they are going to say explain the movement of water or the absorption of water by the root air cell. So the absorption of water by the root air cell is done by osmosis. It is done by osmosis. So osmosis is the movement of water molecules from the region of higher water potential to the region of lower water potential through the selectively semi-permeable membrane. Okay, so now the first question, which is question A, Roman numeral number 1 is saying name the part labeled DP. So part labeled P, it is called D, the root air cell, open bracket, we can say elongated D out growth. So I'm saying part labeled P is called the root air cell, open bracket, we can say elongated D out growth. Then as for Roman numeral number 2 is saying identify substance O. Now this substance O, it can represent the oxygen, or we can say it can represent the water vapor. So I'm going to say water vapor or oxygen. I'm going to say water vapor or oxygen. Then as for Roman numeral number 3 is saying what name is given to the space? What name is given to the space through which substance O comes out of the leaf? Okay, what name is given to the space through which substance O comes out of the leaf? So I'm going to say the name which is given to this space here, it is called the stoma. Okay, the stoma or the stomata. I'm going to say the stoma or the stomata. Then the next one, which is question B, is saying, state the term used to refer to the processes responsible for the movement of water at points L and M. Now, at L, it is showing the movement of water from the root air cell to another cell. Then, the movement of water from the root air cell up to the leaves, it is represented by M. Okay. Now, as for M here, we are going to say it is the transpiration pull. The transpiration pull. And then as for L, it is the osmosis. So I'm going to say the answers here are going to be L is going to be the osmosis. Okay. A is going to be the osmosis. Then as for M, I'm going to say transpiration pull. Then as for question C, it's saying, give 2 reasons why it is beneficial for a plant to lose water at point O. So this one here, this question here, it is asking you the importance of transpiration, because the transpiration here, this is the loss of water. The loss of water by the leaf through the what? The stoma or the stomata. So now, here they are asking you to write the benefits of transpiration. So the first one I'm going to say to cool the plant. To cool the plant. Once the water is lost through the stoma, we are going to find that the plant is going to be cooled. Okay, so I'm going to say to cool the plant. Apart from that one, we are going to say in order to allow the continuous flow of water, to allow continuous flow of water, or we can say sorry, or I may say this one here, the reason why water can be lost through space, which is space, this one, which is the stomata. This is because for photosynthesis, once the water is used, we are going to find, let me say, once water is produced during photosynthesis, we are going, let me say, once water is used during photosynthesis, we are going to find that another water is going to be required. Now, photosynthesis is not going to use the same water. As a result, it is going to use another water which is coming from the root. So I'm saying this one allows the continuous flow of water. Apart from that one, I'm saying it helps it to cool the plant. It helps to cool the plant. Then the next one, which is question D, is saying, apart from the xylem vessel, name another vessel found in the stem used for transport. So I'm going to say phloem, because phloem transport manufactured food. So I'm going to say phloem. Okay. So the next one, which is question 5. So this is the last one on section A. So the question which is question 5, question A, is saying define each of the following terms. Define each of the following terms. So the first one, we have the gene, then followed by the genotype. So the gene, it is just the unity of inheritance. So I'm saying a gene is just a unity of inheritance, or I'm going to say a gene, it is just the alternate form of an allele. So a gene, it is just an alternate form of an allele. A gene is just an alternate form of an allele. Then as for genotype, this one is just the genetic composition of a living organism, or a genetic makeup, the genetic makeup of a living organism, or a genetic composition of a living organism. The way you are made internally, it is genotype. Okay. Usually this genotype, it is represented by the alleles. Genotype is represented by the alleles. Okay, let's move on to the next one, which is question B, the last question. This one is saying, let me say not the last question, we are remaining for one, which is question C. So this one is saying, in a family of rats, the recessive allele, which is small letter D. So recessive alleles are usually represented by small letters. Recessive alleles are usually represented by small letters. Okay. So the recessive allele, which is small letter D, causes standard D growth if it occurs in a homozygous animal. So now when you talk of homozygous, which means on this type on this part here, when we draw our genetic diagram, if we find out that we are having small letter d and small letter d, that animal is going to have standard D growth. Okay, so now as for homozygous allele, if you talk of homozygous allele, they can be different alleles, let me say different alleles with equal expression. Different alleles with, yes, they can be different alleles, or they can be let me say not different alleles but same alleles with equal expression. Same alleles, since homo means same. Okay, homo means same, so these are going to be same alleles with equal expression in the phenotype. These are same alleles with equal expression in the phenotype. Now, usually the alleles are represented by letters. For example, if you talk of small letter d is going to represent a recessive allele. Then when you use capital letter D is going to represent a dominant allele, which means this dominant allele is more powerful. As a result, it is going to show itself in the phenotype. So I'm saying capital letters are represented by dominant alleles. Capital letters are represented by dominant alleles, and dominant alleles are usually expressed in the phenotype, which is the outward appearance of a living organism. Genotype is an outward appearance or an external appearance of a living organism. Now this question is saying, use a genetic diagram to predict the offspring. So the offspring, these are the children. The offspring are the children. So use a genetic diagram to predict the offspring produced if two heterozygous rats were mated. Now, when you talk of heterozygous can be alleles with same letters or different letters. So let me just say heterozygous alleles can be alleles with same letters but different expression. What I mean here is like this. Let's say, for example, you have capital letter D and small letter d, which means these are different. Capital letter D is a dominant allele, and then small letter d is a recessive allele. As the result, capital letter D, which is a dominant allele, is going to show up in the phenotype. Now, when you talk of heterozygous can be, when you talk about heterozygous, we can have different letters, but they are going to have equal expression. You can talk of different letters but equal expression, or you can talk of different letters having different expression. Okay, for example, if you have capital letter A and small letter b, capital letter A is going to be a dominant allele, and then small letter b is going to be a recessive allele. As a result, capital letter A is going to show itself in the phenotype. Now this one here, it is saying we need to use a genetic diagram to predict the offspring produced if two heterozygous rats were mated. So now they have given us the letter to use. So small letter d is going to represent the recessive allele, then capital letter D is going to represent a dominant allele. So the first thing, you need to start with the phenotype, which is an outward appearance of a living organism. So the phenotype here, we can say the male rat close with the female rat. So on this part here, I'm just going to use this paper here. So I will display the answer, okay, like this. So we have the phenotype, which is the male rat close with the female rat. So apart from that one, followed by the genotype. The genotype is an internal appearance or the genetic appearance of a living organism. Then I was saying genotype are represented by alleles, and alleles are represented by letters. So in this case here, since these two rats are heterozygous, I'm going to use capital letter D and small letter d for the male rat, and also for the female rat the same. Okay, then apart from that one, I'm going to split now the gametes. So I'm going to split the gamete. Capital letter D is going to be alone, and also small letter d is going to be alone. Similarly to the female rat, I'm going to split the gametes like the way I was doing on this part here. Okay, so after that one, we need to do the random assortment. Now, during a random assortment, we are going to find that capital letter D on the female rat is going to cut all the values on the female rat. So in this case here, I'm saying this capital letter D is going to go with capital letter D. Okay, capital letter D is going to go with capital letter D, and also capital letter D is going to go with small letter D. So as for the first one here, we are going to have capital letter D and also capital letter D, followed by the next one, which is capital letter D and small letter d. Now on this part here, when you are writing a dominant gene and a recessive allele or a gene, the first thing you need to start with a dominant allele. You need to start with a dominant allele. Don't start with a recessive allele. If you start with a recessive allele, which means you are saying this recessive allele is going to show itself on the phenotype. That's why we usually started with the dominant allele, which is capital letter D. Okay, or the reason why I'm starting with capital letter D, which is the dominant allele. So the next one here, we are going to start with the small letter D to the male rat. So the small letter D to the male rat is going to cut with the all alleles on the female rat. So the first thing here, small letter D is going to go with capital letter D. As the result here, we are going to have capital letter D and small letter D. So here I was saying the dominant allele or the dominant gene should come first, and then also small letter D is going to go with small letter D on the female rat. As the result here, we are going to have small letter D and also small letter D. So this small letter D here has occurred in the homozygous animal. Okay, so we can say small letter D and small letter D, it is a homozygous animal. So now these, they are going to be called as the first filler or first filler genotype. Genotype are represented by alleles and alleles are represented by letters. Then the next one, we have the first filler phenotype. The phenotype is an external appearance of a living organism. So as for D, capital letter D and capital letter D, this one here, it is going to be the normal rat. Then as for capital letter D and small letter D, this one here also is going to be a normal rat, followed by capital letter D and small letter D. This one here is going to be the normal rat. Then the last one, we have small letter D and small letter D. This one here is going to be considered to be a standard growth rat. Okay, so let's move on to the next one, which is the last one, which is question C. So question C is saying, explain one named cause of genetic variation. Explain one named cause of genetic variation. So the first one here, I'm going to say mutation. So now, what is mutation? You need to ask yourself. Here we need to explain what mutation is. So mutation is one of the cause of genetic variation. So mutation, it is the change in the number of chromosomes or in the structure of a gene. Let me say the number of chromosomes or the structure of a gene. The change in the number of chromosomes and the structure of a gene. The change in the number of chromosomes and the structure of a gene. That one is called the mutation. So this one is the